part of genetics today, and we're starting with the karyotypes, which we talked about earlier, which is a diagrammatic representation of the chromosomes. In this case, we can see that they've been stained with some sort of fluorescent dye, which makes it more visible. There's some banding on them that allows us to match certain parts of the chromosome. It's quite visible here on chromosome 12, and we're looking for where the centromere is, the bit that holds the sister chromatids together. Arranged from the biggest to the smallest, and the size of the chromosome generally indicating how many genes it's got on it. And we'll talk about this a little bit more later, but if genes are on the same chromosome, we describe them as being linked, because if you get a gene off chromosome 12, you're going to get all of the genes that are also on chromosome 12. They travel as a pack, they travel together. There's some language on here, and this is the first thing you're going to be asked to do in your worksheet today, and I would be thinking that I would be stopping and starting the video and answering the questions as I go, because it's going to be a lot of information in this first video today. So we've got a picture of the sex chromosomes there, XX in the female, XY in the male, and you can see that the two female chromosomes are bigger than the male. There's some language here. Don't blow your mind straight away. Take your time, think about it, write your notes. It will start to make sense. So you know somatic means a body cell, and in the nucleus of every body cell or somatic cell, in a human, there's 46 chromosomes. And they form 23 pairs, and we say that 22 of them are matched pairs or homologous. That's chromosome 1 to 22, because they're always a matching pair. And a matching pair is called homologous chromosomes, 1 to 22. We also have another word for that. We call them the autosomes. Right, so the autosomes are chromosomes 1 to 22, anything that is not a sex chromosome. Right? So there are autosomes and sex chromosomes. We have 22 autosomes, 2 sex chromosomes. The sex chromosomes are also called heterosomes because they're not always a matching pair, as you can see here in the male. So in humans, that's all in the textbook. Take your time thinking about it, because they are just definitions. You would write XY as a way of saying the sex chromosomes of the male, XX for the female, and you note that the X is larger than the Y. Okay? We look at this a lot better in a lot more detail when we come to inheritance, but sex-linked genes is the term referring to the genes that are carried on the sex chromosomes, on the X or on the Y. It's just linkage, but linkage on the sex chromosomes. It's significant because the X chromosome's got a lot of genes, but it also carries some for rare medical conditions such as haemophilia, muscular dystrophy, and red-green colour blindness. So if you are a girl and you've got one of these recessive genes on this chromosome, you've got the recessive gene for colour blindness here, you are very likely to have the dominant gene here, so you don't have colour blindness. If you're a guy, however, and you've got the red-green colour blindness gene here, you don't inherit another chromosome with matching pairs, so you are going to have that recessive quality expressed. This is what we're going to be looking at in the next area of study. And the Y chromosome doesn't have very many active genes on it. It has got a sequence of genes that is responsible for the male secondary sexual characteristics. All right, but what's most important here in terms of being sex-linked is that sex-linked genes like these are more commonly expressed in guys because they don't have the matching chromosome to hide the recessive gene. Okay, so if you haven't stopped already, you stop now and you go back and do some of the questions, and then you come back to this because this is a different type of thinking and you might find it a little bit complicated. You need to read what's in the book as well as listening to me and looking at the slideshow. Monoploidy we've looked at already. We've looked at this idea of parthenogenesis, where here's the queen who's got 2 sets of chromosomes. Here's her pair of 1, pair number 2. She puts 1 of each into the eggs. This one's been fertilized by a male bee, and we've got 2 sets, a diploid set of chromosomes here, and that develops into a female bee. Curiously, for some organisms, a haploid set of chromosomes is enough for them to develop into a viable organism. And this idea of a haploid set of chromosomes being in every cell of the body of an organism is called monoploidy. This is a monoploid bee because all of its cells are haploid. Again, there's a lot of language, but once you write your definitions, it will start to make sense. All right, so it's monoploid because in all of its cells, it has N chromosomes, just one from each of them. There's not a lot of them. They're in the insect kingdom, and there are a few other organisms that you'll read about in your textbook. Not in humans. It would be lethal in humans. But also it can be a problem because, like with the sex-linked, if this male drone has got a dodgy gene on here, a lethal gene, there's nothing to mask it. There's not a dominant gene to protect it. Okay. Polyploidy. This picture is not properly shown on this slide. I have to go back and fix it, but I'm not doing it now. Polyploidy means instead of having 2 of every chromosome, so in a haploid there's 1 of every chromosome, in a diploid there's 2 of every chromosome. In polyploidy there can be 3 sets or 4 sets or 5 sets or 6 sets, and it's quite common in plants. Right? Polyploidy, the organism's got more than 2 sets of chromosomes. It's advantageous in plants because it can lead to bigger fruit. So here's my maths. They've got 6 sets of however many different chromosomes are in the body. So there must be 7 different chromosomes in wheats, and they have 6 sets of those to give them 42 chromosomes. Tobacco has 48 chromosomes altogether. It's got 4 sets of however many chromosomes. So it must have 12 different chromosomes and 4 of each. And I'm not going to keep going with the maths because I'll embarrass myself. You guys can do that yourselves. It can have a negative effect on fertility, but fruit growers get around that. So strawberries grow asexually, and sometimes they graft the fruit to get around that. Potatoes grow asexually. They have ways of getting around that. And I've put those transitions on twice. Okay, this one's a little bit harder. You come straight to the Google Meet if you've read this and you don't get it. This is normal mitosis. In normal mitosis, we have, in meiosis 1, they line up in the center, the matching pairs, and 1 pair goes to each cell. And in meiosis 2, the sister chromatids separate, and we end up with 1 each of them. Sorry, it's going to keep making that little bar pop up. 1 each of them, N, 1 of each, right? Non-disjunction means that somewhere along the line, one of the chromosomes does something wrong. So if we have a look here at meiosis 1, what's happened here? These 2 lined up in the center, but instead of going to opposite ends, 2 of them went one way. So when we get down here and the sister chromatids separate, instead of there being 2 sister chromatids, on this side we've got an extra one, we've got 3, and on this side they're missing 1, we've only got 1. So if N is the number you're meant to have, N is 2, this has got N minus 1, it's only got 1, and this has got N plus 1, it's got an extra 1. So here there was a non-disjunction at meiosis 1. This is another sort of non-disjunction. It can happen at meiosis 2. Non-disjunction means that they don't separate when they're meant to. So here they do the right thing. They line up in the middle with their homologous pairs, and one goes this way and one goes that way, and we end up with 2 daughter cells with 2 sets of chromosomes. But unfortunately, when the sister chromatids separate, so this one's done the right thing, the sister chromatids have gone to either end, and it's got N equals 2. Here, what's happened? Instead of the sister chromatids separating, they both went to one end of the cell, and the bottom cell here's got 1 extra chromosome, and this one's missing a chromosome. This process of the cell ending up with not an extra set, but just 1 extra chromosome somewhere is called aneuploidy. Okay, so aneuploidy is the presence of an abnormal number of chromosomes in a cell. In meiosis, it would lead to us having 1 less, which would be 45, which would be caused, say, by this one, or 1 more, having 47, which would be this one. Okay, and it leads to certain diseases. So if you don't get that, come straight back to the Google Meet and talk to me. All right, so I thought putting the diagram first might be helpful. Let's see. So this is what we would expect. We would expect diploid. 2 of number 1, 2 of number 2, 2 of number 3, 2 of number 4. In polyploidy, we end up with, instead of 2 of each of them, in this case we've got triplets, and in this case we've got quadruplets or tetraploid. So this is normal for whatever plant this is. This is meant to have 3 of everything, and this is meant to have 4 of everything. That's polyploidy. In aneuploidy, something's gone wrong. So if we have a look here, we're meant to have 2 of this, and we're meant to have 2 of this, but we've got 3. That's not right. And we're meant to have 2 of this, and we're meant to have 2 of this. If this is chromosome number 2, we would say there's trisomy 2. There's 3 of chromosome number 2. What's happened here, we're meant to have 2 of everything. We've got 2 of number 4, 2 of number 3, 2 of number 2, but we're missing a number 1. This is monosomy 1. And this is just the notes. I'm not going to read it. You can come and you can read through that because I just have summed up what was on the slide before. All right, so probably the most common trisomy that we have, so this is a type of aneuploidy where there is an extra chromosome 21. Something went wrong in meiosis. So we look at the carrier type and it's all normal till we get down here and there's 3 chromosome 21s. This is a non-disjunction. At some point, 2 of the chromosomes or 2 of the sister chromatids went into the same egg. In this case, it leads to Down syndrome, and here are all of the symptoms of somebody who's got Down syndrome. This is a viable cell. Having an extra 21, it still develops, but it develops abnormally. Klinefelter's is another syndrome. So if we have a look down here, we can see there's been a non-disjunction either in the egg or in the sperm, where we've ended up with 2 X's instead of 1 X. Because we've got the Y, we should only have 1 X. So we've got 3 sex chromosomes, we're XXY, and you can see on the diagram what some of the symptoms of someone with Klinefelter's syndrome has. All right, I'm not going to read through that because you can read that for yourself. I'm more interested in how the aneuploidy happens. Oh my goodness, sorry about this. Turner's syndrome, there's a little video there for you to watch. Women with Turner's syndrome this is another problem with the sex chromosomes. Short stature, their ovaries don't work, they have infertility, puffy neck, blah, blah, blah. You can read all of that. And again, the double things. And another video, but what's happening with Turner's syndrome? XO, why does it say XO? Because it should say XX. There's only 1 X chromosome. These are female because they've got the X chromosome. They present as female, but they haven't got all of the information they need for proper female development. All right, so it's another issue with the sex chromosomes. And I don't want to go that far. All right, so ignore that last slide. I'm going to make another little video for next week related to that. Not for next week, maybe for next lesson. Good luck with your work today. Please come back to the Google Meet if you need me. It's the only way I can teach you is if you come and ask me for help.